Hi everyone, today I want to talk about something that has revolutionized the world of molecular biology called DNA sequencing. Today we'll be looking at the history, the applications and the workings of these methods. DNA sequencing has truly revolutionized the way we interpret and understand biology. Sequencing has allowed us to determine the identity of organisms as small as bacteria and even to organisms as complex as humans. It has become an essential tool in forensics where DNA found at the crime scene can be matched to a potential murderer. Sequencing has become so critical in everyday life that it has driven technological advancements in such a way that we can achieve the unimaginable. Only 50 years ago, we had to use inaccurate biochemical tests to determine the identity of some bacteria. However, today with the precise sequencing technologies and the user-friendliness, anyone can sequence any organism to find its identity and associated genes and what their function might be. Do not forget the role of sequencing in pathogens. For example, the COVID-19 pandemic going on. The positive news that came from this was the sequencing of the virus's genome. This discovery has opened up so many doors in understanding how the virus functions. For example, where the virus binds on human cells, potential virulence factors, as well as mutation rates, which is what makes a virus very dangerous. And by analyzing the virus's genetic makeup, we can determine whether or not the virus was engineered or is a natural occurrence. Now, let's dive into what these technologies are and what they do. But before we can do that, I believe it's important to go back in the past to see where these methods came from so that we can appreciate the way they work today. Undoubtedly, we've all heard about iconic giants in science. For example, Isaac Newton and Albert Einstein. These are two household names. There are some unsung heroes in the field of biology. For example, Francis and Crick, the two persons who proposed the double helix structure of DNA. Without their pioneering work, we never would have been able to read the structure of DNA, the blueprint of life. The first segment of DNA to be sequenced was that of the sequence for the amino acid alanine by Robert Harley. These findings were published in 1965, and for this he won a Nobel Prize in the field of medicine. I think it must have been amazing to see a DNA sequence that has only been a theory up until that point. The method of sequencing Robert Harley used was quite tedious and complex. It made use of ribonucleases to cleave DNA at specific positions. This would result in a couple of fragments of DNA that had to be puzzled together to be formed in an entire sequence. Although this was a paradigm-shifting discovery, and it was worthy of a Nobel Prize, this did not form the basis of modern sequencing. Now, this breakthrough was made primarily by William Gilbert and Frederick Sanger. Independently, both of these persons developed their own methods for sequencing in the 1970s. Is it just me, or does William Gilbert look exactly like Leonard Hofstadter? These methods were relatively simple without the need for complex puzzling. Ironically, with today's whole genome sequencing, we get quite a lot of data, and it all is sort of all over the place, and this has to be puzzled together. So what made these discoveries by Gilbert and Sanger so user-friendly and so uncomplex was the use of visualizing the DNA sequence on a polyacrylamide gel. Both of these methods relied on fragmentation of the DNA at certain nucleotide positions to determine the sequence. Let's have a look at how these worked. The Maxim-Gilbert method used chemical fragmentation. Briefly, the DNA of interest would be equally distributed in 4 tubes. Each tube would be treated with a chemical designed to break the DNA sequence at a specific nucleotide or 2 specific nucleotide positions. The result of the chemical reaction is DNA fragments of different lengths, and the pattern of breakage, as well in which lane the band is, would correspond with a certain nucleotide. The contents of each tube would be transferred to individual wells of a polyacrylamide gel. After performing electrophoresis, the order of fragment size and their corresponding nucleotide break position would infer the DNA sequence. So this method worked quite well for its time. However, it wasn't very popular due to the labeling of the DNA fragments with a radioactive substance. Luckily, Sanger had an alternative solution. Similar to the Maxim-Gilbert method, the Sanger method would use fragmentation by polymerase reaction termination. This would also be performed in 4 individual tubes. In each tube, a dideoxynucleotide would be added for each of the 4 nucleotides, which acts as terminators that would halt the polymerase reaction. The resulting reaction of each tube would be various lengths of DNA fragments, all ending with the nucleotide of interest. The contents of each tube would be transferred to individual wells on a polyacrylamide gel. Following electrophoresis, the result would entail DNA fragments in 4 lanes in the order of the DNA sequence. Remember that the end of each sequence represents a specific nucleotide. The DNA sequence can be deduced by following the order of the nucleotides based on their position on the gel from top to bottom. So this method by Sanger became known as Sanger sequencing, and today it forms the basis of modern sequencing technology, and it's still used widely today. Just an interesting side note, this Sanger sequencing method was used during the Human Genome Project, which took 13 years to complete. The project aimed to determine the whole sequence of the human genome, as well as different genes that perform its specific functions. Did Sanger and Gilbert win a Nobel Prize for their trouble? Yes, they did. In the 1980s, they won a Nobel Prize in the field of chemistry, shared with Paul Berg for their individual contributions to the world of sequencing. In my opinion, the most important part of any sequencing process is the preparation of the DNA. A successful DNA sequencing reaction requires a good DNA preparation process. Here follows a brief overview of the DNA preparation process for sequencing. First, the sample type and representative species have to be prepared. This is followed by a reliable and efficient DNA extraction procedure. From the resulting DNA, the specific DNA target has to be amplified for sequencing. Finally, this target has to be purified to ensure for the most accurate sequencing results. Depending on the nature of the sequencing method used, preparation of DNA sources is imperative to achieve the desired results. When using high-throughput methods such as Illumina-MiSeq sequencing, numerous species can be sequenced simultaneously. Therefore, DNA can either be extracted from a mixed culture and sequenced, or DNA from various species can be pooled prior to sequencing. For Sanger sequencing, however, it is imperative that a pure species is obtained, because Sanger sequencing cannot distinguish between 2 or more different DNA sequences. This is easy with macroorganisms such as fish. However, in the world of microbiology, numerous bacterial isolation, purification, and culturing would be required to obtain a pure culture. Even with gram staining and microscopy, it does not guarantee a pure culture, which would reflect in the result of the DNA sequence. The outcome of DNA extraction determines the quality and success of DNA sequencing. It is therefore imperative that a reliable DNA extraction method is used without compromising the quality or the structure of the DNA. For example, when we have a mixed culture of bacteria, you would want to be able to obtain as much of the DNA information that there is available. You can't just exclude some bacteria and just decide not to account for that DNA. So therefore it's important when using a correct DNA extraction method to lyse as many cells as possible so that you can obtain a representative DNA sample. If done incorrectly, this will lead to a bias where some bacterial species will be sequenced and some not, and this will be a misrepresentation of your original sample. A DNA extraction protocol consists generally of 4 parts. First, it's cell lysis, separation of DNA from cellular debris, DNA purification, and dilution of DNA in a liquid, usually a buffer. If a mechanical lysis method is used, in other words, if a physical method is used to break open the cells to expel the DNA, this could have some dire consequences for your DNA, and it will result in some extreme breakage. And this will lead to the inaccuracies when sequencing, or if it's sequenced at all. Now, the purification of the DNA is equally as important. If the DNA is not properly purified from contaminants, it will have an influence in downstream reactions. The most notable of these reactions is PCR, polymerase chain reaction, and which is an essential component for Sanger sequencing. And a final consideration for DNA extractions is the elution buffer in which the DNA is finally stored. Usually a buffer would be used because it prolongs the stability of the DNA for long-term storage. However, if these buffers contain EDTA, the EDTA itself might inhibit downstream reactions such as PCR. However, if nuclease-free water is used, it might shorten the life of the DNA's integrity. With the exception of whole genome sequencing, whether you are trying to sequence a target sequence from a mixed or pure culture, the sequencer cannot guess which part of the genome is important to you. For example, when identifying bacteria, we usually use the 16S gene, which is unique to different bacterial species. This fragment is about 1,600 base pairs long, whereas the genome of a typical bacterium is around 4.6 million base pairs. What needs to happen next is we use a process called PCR to identify that fragment of 1,600 base pairs and amplify it so that it can be sequenced individually from the genome. You can think of PCR as a method of copy and pasting a fragment of DNA. Now, this happens exponentially, which means if one initial fragment exists and we take that fragment, we copy and paste, then we have 2 fragments. The next cycle, the 2 fragments become 4 fragments. The next cycle, the 4 fragments become 8 fragments, and so on and so forth for usually about 35 cycles. In the end, this will result in about 30 billion copies of the original fragment you want to sequence. How we specifically identify a sequence on an entire genome is with the use of primers, highly specific pieces of DNA designed to bind complementary on the genome. The binding of the primers to a genome mediates the attachment of a polymerase enzyme which builds the DNA sequence exponentially over a fixed number of cycles. So to summarize, we attach primers specifically to a sequence that's important to us, it becomes amplified, and we use that amplified product to sequence specifically. Now the final step to prepare your DNA before sequencing is to purify your PCR product. Your PCR product consists of the DNA you amplified, but it also consists of other things that might contaminate downstream reactions. This includes nucleotides and primers. So what this purification step does is it removes all these contaminants from your amplified DNA product. It's very important to remove these contaminants because for sequencing, you accurately need to determine the quantity and quality of your DNA. This cannot be done with residual nucleotides within your DNA mixture. If you were to quantify your DNA without purifying the product, that is to say, it still contains residual nucleotides and primers, it will lead to a false measurement of your DNA quantity. This will surely influence your sequencing reaction. Once we have completed the preparation of our DNA, we can determine the quality and the quantity of the product. The quality of the DNA can be assessed by running agarose gel electrophoresis. The quantity can be measured using machines such as the nanodrop or qubit. Determining the quantity of DNA is very important for sequencing because sometimes we use up to pictograms in mass. As mentioned in the previous sections, Sanger sequencing formed the basis of modern sequencing. However, this does not mean that the method is outdated or no longer used. In fact, it's probably one of the most common sequencing methods used today. Considering the popularity of this method, it's imperative that molecular biologists understand this method, how to interpret the results, and its potential applications. Only now do we get to the sequencing method itself. Remember that Sanger sequencing uses fragmentation to determine the DNA sequence. However, 2 developments improved this method substantially. These improvements made the sequencing so much more user-friendly and doable within a reasonable amount of time. The first development was the use of dideoxynucleotides that were fluorescently labeled. This allowed for a machine to detect the fluorescence of each individual nucleotide and thereby the nucleotide sequence. Secondly, since the machine can distinguish between nucleotides based on their individual fluorescence, the sequencing reaction could take place in 1 tube instead of 4, which significantly streamlined the preparation. The first step of this process is to create fragments of different sizes of the target DNA. This is performed using a PCR process. However, the PCR reaction is not conventional. The first major difference is by only using 1 target-specific primer, either forward or reverse. The sequencing reaction PCR is prepared, which contains buffer, the forward or reverse primer, target DNA, and individual nucleotides. These nucleotides consist of a ratio between normal dNTPs and fluorescently labeled ddNTPs. During the PCR reaction, complement sequence is constructed using random NTPs to construct the sequence. In some cases, the normal dNTPs will be used and less frequently a ddNTP will be used. The ddNTPs have 2 features: a fluorescence tag unique for each nucleotide and a PCR terminator. So when the ddNTP is added to the sequence, the PCR reaction for that sequence is halted, resulting in a DNA fragment with the last nucleotide of the sequence labeled for detection. A fragment for each nucleotide position is synthesized. thus resulting in DNA fragments of various lengths. Following the PCR reaction, the DNA is not visualized on a polyacrylamide gel. Rather, the DNA is purified in a second cleanup step. This step is imperative since it removes all excess dd-NTPs. Since the sequencing machine measures fluorescence, excess fluorescence and unnecessary fluorescence will influence the sequencing results. The sad truth is that all a Sanger sequencer does is measure the fluorescence of nucleotides to determine the sequence. This process is called sequencing by capillary electrophoresis. The DNA fragments are sent through a capillary tube in which the DNA is separated based on size. Very similar in principle to conventional electrophoresis. However, the capillary tube only allows one DNA fragment to pass through the tube at a time. Smaller fragments move through first until it reaches a light detector, which measures the emission of fluorescence. Since each nucleotide emits its unique fluorescence, it can translate the exact nucleotide. As the fragments move through the tube from small to large fragments, each fluorescence signature is measured, translated, and added to the previous measurement, eventually creating the DNA sequence. Alright everybody, we are finally at the end of our video. I really hope you enjoyed it and learned something. This video is not for everyone, it will be for persons coming from their undergraduate studies to their postgraduate studies. However, if somebody else watched this, I hope they learned something about the intricacies of DNA sequencing. If you have any questions, do not hesitate to ask. If I cannot give you the answer, I will refer you to someone who can. So, stay safe, stay smart, and thank you for watching.